Well, thank you, Joan, and thanks for the invitation. And I've got some people here that helped me organize this talk, provide information for the talk, and I also want to thank, as Lindsay thanked, the Sloan Foundation for support, and also the Berkeley Indoor Microbial Ecology Research Consortium, which was funded by the Sloan Foundation. When I got this invitation and then started to think about the talk, of course looked at recent literature, and then I also looked at who's on the panel, and there's a remarkable concordance there of people who are really active now and are on the panel. It's a very good panel. And it made me wonder, what could I possibly contribute? And then in my searches, I found this really nice recent review by Brent, which is a talk that he gave to the panel, and it made me even more concerned. And so I was concerned, but then deep in the report, Table 1, there's a statement here. Fungi mostly come from outdoors, while bacteria are mostly emitted indoors. And that is at odds with what we've discovered in our work at Berkeley, so I thought that would be what I'd start with. And I'm going to talk about fungi indoors and outdoors, and then I'm going to say something about bacteria indoors and outdoors, and then finish with practical implications, about a third of the time for each one. So when we started this, Tom Bruns, who led the project, is an ecologist, and his mantra was replication and sampling. And the idea was to reduce the number of variables, so we picked units in married student housing at Berkeley that have the same floor plan and were built in roughly the same time period. And then to sample, we developed a very inexpensive sampler. This is a large distance shot, and here's a close-up. And the apparatus is just a Petri dish lid hung from something. We started with strings, and this is strapping tape. And then that's a sterile Petri dish next to it, and you just take the bottom out and invert it and put it in that holder and leave it for a few weeks. And then you come back with a new sterile Petri dish, and you put a sterile lid on it and label it and take it home. And then do the same sorts of analysis that everyone's talked to you about. And when we did that at the married student housing, we had 12 units. We then tried to correlate, or wanted to correlate what we found with the unit, the distance between the units in essence, and then the room type, the age of the structure. There was 3-year difference in some of the units. The flooring, the number of bedrooms, bathrooms, and then the inhabitants, the occupants, and how many and what they were doing. And also plants. There are no pets allowed in this area. And when we did that analysis, we found the only significant correlation was between the units, or the distance between the units. And that all of those things inside, the room type, this is for fungi, the occupants, how often they cleaned, what they cooked, really didn't matter. And so we wrote that geographic distance, and the proxy for that unit, is the single significant determinant of community structure, and not, for example, room function or resident behavior. And you can see even in this small married student housing area where the distances are about half a kilometer, you could see that the community, the fungal community difference correlated with the geographic distance difference. In other words, there's a limitation to dispersal for these fungi, or distance decay. So for fungi, the indoor air, the fungal cohort in a well-maintained building is filtered from outdoors. And fungal dispersal is limited from a few meters, as in this area next to Berkeley, up to halfway around the Earth when people look at global studies. Now, I'm talking about air today. I'm not going to talk about surfaces. And fungi, in terms of the air and also most of the surfaces in the house, our feeling is fungi live in a desert. It's punctuated by some oases, and that would be, for example, the drain in a sink. It would be a very different fungal community where there's a lot of water. But in the air and on the surfaces, no. So then we had the DNA from the fungal study, and we had primers for bacteria, so we took a look at the bacteria. And here's a graph from that, and it starts here. This is the balcony. This is our outdoor sample. And then the 2 rooms close to the balcony that are off the balcony are the kitchen and the living room, and then deeper into the unit, the bedroom and the bathroom. And you can see that there's an effect as you go from outdoors into the kitchen and the living room, and then as you go deeper into the unit, where there's more intimate association with the inhabitants, the bedroom and the bathroom, there's the community of bacteria changes more. And so we wrote, while fungal composition was structured by season and residential unit, fungi vary in the seasons, bacterial composition varied by residential unit and room type. So there is a human contribution, certainly in bacteria. The question is how much. And for the bacteria, we also saw this distance decay, that the community of bacteria, the distance between those communities in the different units also correlated with geographic distance between the units. And so bacterial communities indoors in well-maintained buildings are dominated by a combination of outdoor bacteria and human-associated bacteria, and communities of bacteria show dispersal limitation locally as well as globally. So this points out that there's a human contribution, but doesn't talk about how much. So then we went from descriptive studies to experimental studies, and the architecture school at Berkeley has a chamber. It's a 5-meter by 6-meter room, 2.5 meters tall. It's got some fans on the ceiling to mix the air. And we sampled the air here instead of the passive sampler because the experiments lasted for 2 hours. We had a vacuum pump that took 25 liters a minute for 2 hours. So we sampled about 3 cubic meters of air, 3,000 liters of air. And the variables were occupants. We'd have unoccupied, 1 occupant, 2 occupants, or 8 occupants. Activity, when we had 2 occupants, they'd either sit or in a different experiment, they'd walk around. And then the floor, it's either exposed or here you can see it's covered. And so from these studies, I should mention one other thing. And we had another one of these units outside the building. So our conclusion is the microbial communities observed in the indoor air samples largely tracked those simultaneously measured outdoors. Taxa known to be associated with the human body played a secondary, although important role. These would be bacteria. And from sequence read abundance, a modest enrichment, 1.3-fold, was observed indoors at 4.3% of these types of bacteria versus 3.7 outdoors for the 5 human skin taxa listed here. And then from the frequency of taxon occurrence, 8% of the 134 bacterial taxa showed increased frequency with increased occupancy, most of which were environmental rather than skin associated. And so like Lindsay, we suggest that resuspension of outdoor-derived microbes from indoor surfaces or from occupants' clothing was a stronger source than direct shedding from human bodies. And so these results led Rachel to make this cartoon in which you have air coming in, bringing microbes in, exhaust, microbes leaving, deposition due to gravity, and then the effect of the humans, resuspension is a strong effect. We believe transport on clothing and surfaces is an effect, and then shedding of human-associated microbes is significant, but not a major effect. Now, we're not the first people, the only people, nor the first people to come to this conclusion. So here are a couple of papers from Jordan's lab. And there they say a significant proportion of the bacterial emissions, 18%, were distinctively associated with the human microbiome. And that indoors, taxa that are unique to human skin were found at a total enrichment of 17%. Outdoors, the total enrichment of human skin, hair, and nostril microflora was 4.9%. So they see a 3.5-fold increase in indoor versus outdoor. And then also from the Oregon group, from Jess Green's group, there's a study, and they conclude indoor air communities closely tracked outdoor air communities, but human-associated bacteria genera were more than twice as abundant in indoor air compared with outdoor. So they see a 2-fold enrichment indoor for bacteria. So there are a number of such studies, and they've been tabulated in a very recent review that many of you are co-authors on. And Rachel shared this with me. I don't think it's out yet. And here's table 1. And to make the title larger so we can read it, percentage of sequence in indoor air studies derived from human sources. And then the last column has these numbers, and I've enlarged it too. And you can see that it goes from a low of about 4% to a high of 32%, with most of the other ones in between. That 4% is our chamber study. The 32% was our married student housing study. So it's important, but it's not the main source indoors. So I would take issue with Brent's statement and say fungi mostly come from outdoors, while bacteria are mostly emitted indoors. Instead, I'd say fungi and bacteria come from outdoors. In addition, bacteria are emitted indoors. So now I want to go on to the practical implication part. Start with litigation. And now we're moving from well-maintained buildings to buildings that are potentially poorly maintained. So many of you have probably seen this. This is a mold report that you'd get if you consult on a legal matter. And in this case, the sampling was 5 minutes of sampling with a vacuum pump that could handle 15 liters per minute. And up at the top there is a key that tells you what the various samples are. And the last sample here is the outdoor. They're very good about having outdoor and indoor. And then here's a list of taxa. These are fungi, some to genus and some a little broader. So for example, Aspergillus Penicillium is a group that would take in about 600 species, and then ascospores probably about 70,000 described species, and basidiospores about another 40,000 described species. So that they're not nearly as precise as what we're able to do in the field now. And then you wonder how much did they sample, and I told you that they sampled 75 liters of air. You can tell that by looking at the number of spores estimated to be in a cubic meter and dividing 1,000 liters by 13, and you'll get that 75 figure as well. Or you can look at the bottom where they give you the parameters of their sampling, and you can see that there's the flow rate, the 15 liters per minute. And so I said that they sampled 75 liters, but if you look up here, they say volume analyzed is only 18 liters, or 18 thousandths of a cubic meter. And the reason for that is that they didn't count the whole slide. These are spores that are impacted on a microscope slide, and the technician looks at the slide. And so they stopped after looking at a quarter of them. So they really only analyzed 18 liters worth of sample. And at 15 liters a minute, that's about a 1 minute sampling of air at one point. And huge amounts of money exchange hands as a result of this. And I think we know in this field that's not enough sampling. So now I want to go to medicine, and Mark's going to talk to you this afternoon. But this is a really nice review. It's a sort of a meta-analysis of asthma and exposures to microbes from last year. And in this, they come to many conclusions, but the one about fungi that has statistical significance is there's sufficient evidence, that means statistically significant evidence, of a causal association between outdoor culturable fungal exposure and exacerbation in asthmatics sensitized to fungi. And then later in the paper, they're very careful. They point out an important weakness of these studies on airborne culturable fungi is that all those specifying their sampling methods used 1 minute air samples and thus had a highly unreliable estimate of fungal concentrations. So again, we can do better than that. And people are doing better than that. So this is again from Jordan's lab. And they've been looking at asthma and indoor microbes, and they have a visit at the start of the study, and they get a 3-minute collection vacuum sample from furniture and a 2-minute collection from a 1 square meter of floor. But again, that's different than the sampling that I talked about with the passive samplers, because it's not over a defined time period. And that is one problem with the vacuum approach or the swab approach, is that you have an undetermined length of time. So now I want to go to poorly maintained buildings in the last bit. And this is Iman Sylvain, graduate student at Berkeley, and a poorly maintained building, which is public housing. And I want to show you the roof of the building. I mean, you can see that it's had many patches, but I also want to point out that the roof does go over the entire building, including the overhang on the open hallways. And this is sky. This is looking up at one of these at the top of the building. So this is the overhang over the exposed walkway. And that is a stalactite. That is dissolved concrete that has formed a stalactite. And directly underneath it is a stalagmite. So this building has been poorly maintained for a long time. And so we put out our samplers, and this is outdoor, and this is like a principal components analysis of the communities. And you can see that's a fair approximation of the outdoor air at this area. And these other ones that are outliers are possibly due to our placement of the outdoor samplers. I think we unknowingly placed some near the kitchen exhaust fans. And now here is, we still have the outdoor, and they're added the indoor. And you can see that many of these are like the outdoor air. So those are units that don't have, that are well maintained enough that they are just filtering the outdoor air. But you can see there are many that are not. And if you look at some of those and look at the fungi involved, you can see the blue ones are common outdoor and filtered. There are fewer of them indoor. So the building is doing its job on the outdoor fungi. But you can see that there are many that are much more common indoor than outdoor. And so these are likely generated within the structure. And so I want to go back to the sampler, that the key thing here is sample amount, and it has to be inexpensive if we're really going to have an impact on health or on litigation. And so something like this, where it's very inexpensive and it samples over a defined period of time, I think, I want to make a pitch for, I think that is the way to go. And with that, I'll stop, and I want to thank people, especially Paula and the Sloan Foundation, for launching the field, or at least launching me into the field, not launching the field. And then the people at Berkeley, I mentioned Rachel and Iman and Tom Bruns, and Seema, who was also a postdoc. And then these are the PIs on the BIMERC grant. Thank you very much.